Which are the already visited, that should be ignored. Which is already visited, that to be ignored. Already A to be visited, that is the reason B to A not visited, already visited. Now B, is there any B neighbors? No. B neighbors, 3 neighbors, A already visited, C and D, it is completed. Next I want to continue for order wise. A neighbor, first neighbor B, B neighbors I continue. Then I want to go for E neighbors. E neighbors, first in first out, first priority goes on first. E neighbors, what are the E neighbors? A, F, D. A neighbors, A, D, F. E to A, yes, E to A visited already. E to D, no. E to D not visited. E to F, no. Now which to be visit here? I want to visit that E to D. E to D, D to visit. E to D. Now this D already visited, is it right? Here B to D visited, here E to D I want to visit, E to F. E to F. After that E neighbors completed, then I want to move on that F. F to be visit, F neighbors. Coming to the F neighbors. Now I want to continue for F neighbors. Which are the F neighbors? This is my F. What are the F neighbor? A, E. F to A already visited? Yes, A to F. Is it E to F already visited? Yes. Now F is processed state. F already 2 neighbors, that is already visited. E and F already. F neighbors are A and F, both are visited. Now A and E both are visited. Now continue for the F is completed, then go for B neighbor. What is the B neighbor? C. C to be start, C. What are the C neighbors? Now I want to visit for C neighbors. What are the C neighbors here? C.
Hello students, today in this presentation you people are going to learn the topics DNA as genetic material, DNA structure and DNA replication. First, I will start explaining the topic DNA as genetic material. To get it studied, it is better to introduce the knowledge of how and why a nucleic acid like DNA was considered as the genetic material. This is followed by the experiments of Griffith, Averroes, Hershey and Chase experiments. So first let us come to know the introduction part. In the first half of the 20th century, Gregor Mendel's principles of inheritance became widely accepted. But the chemical nature of hereditary material remained unknown. Based on his experimental investigations, Mendel concluded that development of characters were due to the presence of particular factors which were called as genes, and these particular factors were transferred from one generation to another generation. However, the chemical nature of genes fascinated the scientists for many years. But during 1920s, a series of experiments conducted by scientists finally revealed that DNA was the genetic material. But here we have a question: what decide which molecule can be a genetic material? There were certain properties. Whichever molecule satisfy these properties can be a genetic material. So let us come to see the properties of genetic material. These are the properties of genetic material. The hereditary information must be there in the structure of the genetic material. It should undergo replication. It should be able to express itself through the formation of specific biomolecules, and it should be able to undergo mutations, and such a change should be stably inherited to the next generations. Having seen the properties of genetic material, we will now focus on Griffith's experiment. Regarding this, it was in 1928, Frederick Griffith conducted a basic experiment on bacterial transformation. In the process of identification of DNA as the genetic material, Griffith conducted experiments on Diplococcus pneumoniae, a bacterium which causes pneumonia, and this is also known as Streptococcus pneumonia. In his experiments, Griffith used two strains of Streptococcus pneumonia. One is called S3 strain and the second one is R2 strain. Coming to know the properties of S3 strain. These bacterial cells have smooth polysaccharide capsule and they are capable of producing smooth colonies. It is a virulent bacterium. That means it is able to cause pneumonia. Because of the presence of this smooth polysaccharide capsule, it is protected in the host immune system and able to cause pneumonia. When it comes to R2 strain properties, these bacterial cells don't have smooth polysaccharide capsule. They are capable of producing rough colonies. It is a non-virulent strain. Non-virulent means non-pathogenic strain. They are unable to cause pneumonia. Because of the absence of this smooth polysaccharide capsule coat, they will be damaged in the host immune system and unable to cause pneumonia. During the course of his experiments, Griffith injected mice with S3 strain. Mice developed pneumonia and died. Then, when mice were injected with the R2 strain, there were no signs of pneumonia because it is a non-virulent strain. Then, he killed S3 strain by subjecting it to heat treatment and injected into mice. There were no symptoms of pneumonia, indicating that all the cells were killed by heat treatment. Then he tried a combination of heat killed S3 strain and mixed them with live R2 cells and injected into mice. Surprisingly, there were symptoms of pneumonia. Here we get a question: if it is a heat killed bacterium, will it be able to cause disease? The answer to this question was like this: analysis of dead mouse tissue was found to contain live R2 cells which had smooth polysaccharide capsule like S3 cells. This is the diagrammatic representation of Griffith's experiment. And what did this experiment prove? And what must have gone into the R2 cells? From the results, Griffith concluded that there was something that was transferred from heat killed bacterium into R2 cells which helped them to synthesize a smooth polysaccharide capsule and became virulent. This was called as bacterial transformation by Griffith. That means one bacterium was changed into another bacterium. So here non-virulent bacterium was transformed into virulent and the substance that was transferred from virulent bacterium into non-virulent bacterium was known as transforming principle by Griffith. So today we know transformation is a specific type of recombination. A segment of DNA replaces another homologous segment on bacterial chromosome. However, Griffith's experiment was a turning point towards the discovery of DNA as the genetic material. But he could not identify the biochemical nature of transforming principle in his experiments. Now, the focus was on finding out the biochemical nature of transforming principle in Griffith's experiments. Many scientists performed different experiments, there were different observations, results and opinions. A team of scientists, Avery, McCall and McCarthy, performed experiments to determine the biochemical nature of transforming principle in Griffith's experiments. So for this, they first extracted various biochemicals like carbohydrates, proteins, lipids, RNA and DNA from heat-killed S3 strain. Then these extracts were subjected to treatment with different enzymes and alcohol before they were mixed with live R2 cells and injected into mice. The observations and results were recorded like this. First, the cellular extracts were mixed with alcohol. They observed that it got precipitated by alcohol. In the second part of the experiment, the cellular extracts of S3 strain were subjected to treatment with protease enzymes, lipases and RNases and injected into mice. It could cause transformation. That means mice developed pneumonia and died. Then the scientist subjected the cellular extracts of S3 strain with DNase enzyme and mixed this preparation with live R2 cells and injected into mice. It could not cause transformation, indicating that the transforming ability was lost. Then what could be the transforming principle? It was confirmed by the researchers it was not a carbohydrate, not a protein, not a lipid, not RNA. How could they confirm this? Being a carbohydrate, it would have not got precipitated by alcohol. So, it is very clear it was not a carbohydrate. Proteases are enzymes with which proteins will get damaged. Lipases are enzymes which destroy lipids. RNases are enzymes which destroy RNA. But even in the absence of these biomolecules, transformation happened. So it is very clear that these biomolecules were not responsible for transformation. Then the scientists were left with one option to decide which biomolecule is responsible for transformation. It was DNA. After treatment with the DNase enzyme, transformation could not happen. There were no signs of pneumonia in mice, indicating that the transforming principle was lost. Then what did this experiment provide? What did this experiment show? The genetic material that is getting passed on from one bacterial cell to another bacterial cell was nothing but DNA. So DNA can transform the properties of a cell. Here in this case, it was the appearance and pathogenicity of a bacterium. And the results of these scientists were published in 1944. But this experiment was not accepted by most of the biologists due to the lack of information on the molecular mechanism of transformation. So finally, a confirmatory report came from the experiments of the scientists Hershey and Chase in 1952. Hershey and Chase, this is also known as Hershey and Chase blender experiment. They took T2 bacteriophage, which is a virus that infects the bacterium E. coli, Escherichia coli. You have to remember one point, T2 phase itself has only two things in its structure, DNA and protein coat. During the infection of bacterial cells, T2 phase injects only its DNA into the bacterial cell, but the protein coat always remains outside the host cell. And inside the bacterial cell, the protein, the DNA molecule undergoes replication and forms new viruses. This is the life cycle of T2 phase. So remember this. In their experiments, Hershey and Chase had grown T2 phases in two different mediums containing two different types of radioisotopes. In the first sample, T2 phases were grown in medium containing sulfur 35 radioisotope to label the proteins of viruses. In the second sample, the bacterial cells, the viruses were grown in medium containing phosphorus 32 radioisotope to label the DNA. This is the experimental setup of Hershey and Chase. So here it clearly shows that in the first part of the experiment, T2 phages were grown in medium containing sulfur 35 radioisotope to label the protein codes of the viruses. This happens because sulfur is a component of certain amino acids. So it is incorporated into the proteins during protein synthesis. When these sulfur 35 labeled protein viruses were used to infect these E. coli bacterial cells, the protein codes were left outside the host cell. That means radioactivity was left outside the host bacterial cells. Then the non-labeled DNA undergoes replication and forms new viruses. Now coming to see what is happening in the second part of the experiment. In the second part of the experiment, T2 phages were grown in medium containing phosphorus 32 radio-labeled viruses. Because phosphorus is a component of DNA, so it is incorporated during DNA synthesis. So the nucleic acids DNA is labeled with phosphorus 32 radioisotope. When these P32 labeled viruses were used to infect E. coli bacterial cells, radioactivity in the form of DNA enters the host bacterial cells, but the non-labeled protein codes were left outside the host cell. Now within the host cell, these radio-labeled DNA undergoes replication and forms new viruses. After infection, viruses and bacterial cells were separated by blending and centrifugation and then observed for radioactivity by the scientists Hershey and Chase. Now have a look at this slide. Bacterial cells, you can see bacterial cells were formed as a pellet at the bottom of the test tube when treated with phosphorus 32 radio-labeled viruses. That means radioactivity was inside the bacterial cells. But in case of viruses that were treated with sulfur 35 radio-labeled viruses, radioactivity was outside the host cell. That means smaller virus, after centrifugation, smaller viruses remained in the supernatant, but the bacterial cells formed in the bottom of the centrifuge tubes. Now I am talking about the radioactivity. The radioactivity was outside the bacterial cells when treated with sulfur 35 radio-labeled viruses, but it was inside the bacterial cells when treated with phosphorus 32 radio-labeled viruses. So coming to know the conclusion, results of this experiment. What did this experiment provide? This experiment provided a strong evidence that DNA was the genetic material in T2 phases. How can the scientist explain this? How could they explain this? It can be explained like this. During infection of E. coli bacterial cells, all the protein codes were left outside the host bacterial cells. They didn't enter the bacterial cells. So that means they were not responsible for the formation of new viruses. But it was only DNA that entered the host bacterial cells, and as it is having the genetic information of T2 phases, it was transferred from one phase generation to another phase generation. So this way, the findings of Hershey and Chase led to the universal acceptance of DNA as the genetic material. Next, I am going to talk about DNA structure. The things you will study here are introduction, key components present in DNA structure, Watson-Crick double helical structure of DNA. Let us come to see the introduction part about the discoveries of DNA. We have seen how DNA acts as the genetic material, but still there are many questions to be answered, like how DNA stored the genetic information and how is DNA copied so easily. These questions would be answered by discovering the structure of DNA. DNA or deoxyribonucleic acid was first identified by a Swiss chemist Frederick Miescher in 1869 in the nuclei of human white blood cells. It was about in 1953 James Watson, Francis Crick, Maurice Wilson, Rosalind Franklin, using a powerful technique called X-ray crystallography, figured out DNA as a double helical structure. It is a twisted ladder, and they revealed that this structure could carry the biological information. In 1962, Watson-Crick, Maurice Wilson received the Nobel Prize for their discoveries concerning to the molecular structure of DNA, but unfortunately Franklin died of cancer 4 years before these researchers received the Nobel Prize. So here at this point we will discuss the structure of DNA. DNA is composed of 2 polynucleotide chains. Each polynucleotide chain is made up of monomeric subunits called nucleotides. Now let us have a look at the structure of nucleotide. Nucleotides are the building blocks of DNA. Each nucleotide is made up of 3 components: a 5-carbon sugar, a pentose sugar, 1 or more phosphate groups, and a nitrogenous base. Now we will move to discuss the properties of pentose sugar. It is a deoxyribose sugar. The oxygen atom present at carbon atom 2 of ribose sugar is missing, so it gets the name deoxyribose sugar, and DNA has the name deoxyribonucleic acid, and it is attached to the 3' carbon and 5' carbon of this pentose sugar are attached to a phosphate molecule to form a phosphodiester bond, and the 1' carbon is attached to a nitrogenous base by means of a beta N glycosidic bond. It is an inorganic phosphate in DNA structure. It has the formula H3PO4 and it contains 3 reactive groups, 3 reactive hydroxyl groups. 2 of them are involved in the formation of sugar phosphate backbone in DNA. Why we know DNA as an acid? Acidic nature of DNA is due to the presence of this phosphate group. Here we have the structures of deoxyribose sugar and phosphate group. You can see oxygen atom present at carbon atom 2 of this deoxyribose sugar is missing. This property is important for the enzymes to recognize DNA and RNA in the living cells. This is inorganic phosphate having 3 reactive hydroxyl groups. 2 of them are involved in the formation of sugar phosphate backbone in DNA that we will discuss in the next slides. Next, coming to know the nitrogenous bases in DNA structure. Different types of heterocyclic nitrogen ring containing compounds are present in DNA. These are of 2 types, pyrimidines and purines. Now we will discuss about pyrimidines. It is a single ring containing compound and purines are of 2 types in DNA, thymine and cytosine. Thymine is a 2,4-dioxy-5-methyl pyrimidine, whereas cytosine is a 2-oxy-4-amino pyrimidine. Here we have the structures of cytosine and thymine. They are read in clockwise directions and they are similar to benzene ring except that it is having nitrogen at positions 1 and 3. Coming to know the structures of purines. They are double ring compounds containing a 6-membered pyrimidine ring and a 5-membered imidazole ring. Purines, they are of 2 types in DNA, adenine and guanine. Adenine is a 6-amino purine, whereas guanine is a 2-amino 6-oxy purine. Now have a look at the structures of adenine and guanine. Both are double ring containing compounds and you can see that they are fused at positions 4 and 5 carbon atoms and they are having nitrogens at positions 7 and 9. They are read in anti-clockwise direction. Once we know the structural components in DNA, we must have to focus on the differences between nucleosides and nucleotides in DNA. Nucleoside is formed when a deoxyribose sugar joins to a nitrogenous base, whereas a nucleotide is formed when a nucleoside joins to 1 or more phosphate groups. So obviously there are 4 types of deoxyribonucleosides in DNA. These are deoxyadenosine, deoxyguanosine, deoxythymidine, deoxycytidine, and the nucleotides in DNA are deoxyadenilic acid, deoxyguanilic acid, deoxythymidilic acid and deoxycytidilic acid. Naming of nucleotides is based on the presence of number of phosphate groups. Here this is a nucleotide structure and it is having only 1 phosphate group, so it is named as deoxynucleoside 5' monophosphate DNMP. If it contains 2 phosphates, it will be named as diphosphate. If it is having 3 phosphates, it will be described as triphosphate. A nucleotide joins to another nucleotide by means of a phosphodiester bond. A phosphate molecule links the 3' carbon and 5' carbon of adjacent sugar rings like this. This is one type of condensation reaction and two hydroxyl groups of phosphate molecule are involved in this bonding. So in this way this results in the formation of a dinucleotide. So like this number of nucleotides are joined together to form a polynucleotide chain. So this polymer is what we know as DNA. Now coming to know Watson-Crick double helical structure. Watson-Crick suggested that DNA is composed of two polynucleotide chains, two strands of nucleotide chains that are twisted around each other to form a double helix. It is a right-handed helix. That means if you hold it like this, it will they will twist around each other to form a double helix in clockwise direction. It is referred as B form. The two strands of DNA molecule run in opposite direction. That means they are antiparallel. One strand runs in 5' to 3' direction, the second strand in 3' to 5' direction, and the two strands are held together by hydrogen bonds that are formed by complementary base pairs. The base pairing in DNA is very specific. If one strand has a purine base, the opposite strand will have pyrimidine base. This follows Chargaff's rule, and the bases are joined by hydrogen bonds like this. Adenine pairs with thymine by means of two hydrogen bonds. Guanine pairs with cytosine by means of three hydrogen bonds. How exactly does this take place? This is due to the spatial configuration and arrangement of atoms in each base, so that the bases will pair only in certain combinations, and this is helpful to maintain a fixed distance between the two strands, and this allows something called complementary base pairing in DNA. That means both strands are not identical. This is Watson-Crick double helical structure of DNA. It is looking like a spiral staircase with the complementary base pairing, major, minor grooves, and showing diameter and other properties that we will discuss in the next slides. This structure is showing how sugar phosphate backbone is projecting outside of the DNA molecule, and complementary base pairings are stocked inside the DNA molecule. Because of the presence of these large number of hydrogen bonds, there is a high degree of stability in DNA molecule, and these properties are important concerning the mechanism of replication in DNA. And the order of these sequences, base sequences, determine genetic code in DNA. These are other properties of the double helix. Diameter is 20 angstrom equal to 2 nanometer. It has a pitch, that means length of that helix to complete one turn is 34 angstrom, that means 3.4 nanometer, and there are 10 base pair per each turn of the helix. The distance between each base pair is 3.4 angstrom equal to 0.34 nanometer, and the helix has major and minor grooves which are formed as a consequence of arrangement of bases. What is the importance of DNA structure? From this, it is clearly understood that DNA stores the genetic information, it undergoes replication, transfer the information to the next generation, and expressed through the formation of specific biomolecules only because of its unique structure. Next, I will be talking about DNA replication. Here, you will come to know the definition of DNA replication, key enzymes involved in replication, and the mechanism of DNA replication. What is replication? Replication is the process of making copies of DNA. The purpose of replication is to produce two identical copies of DNA. And we know DNA regulates the activities of a cell because of its unique structure. So when the cell divides, DNA should also duplicate to be able to get distributed equally to the daughter cells. That means the genetic material must undergo replication. What are the types of replication? 3 types of replication have been proposed at the molecular level: conservative, semi-conservative and dispersive. But it was experimentally proved by Messelson and Stahl experiment DNA replication was semi-conservative. But we are not concentrating on this experiment. We are mainly focusing on the mechanism of DNA replication. If replication is conservative, one daughter molecule will have 2 whole strands and the second daughter molecule will have 2 newly synthesized strands. If replication is semi-conservative, each daughter molecule will have 1 parental strand, that means 1 whole strand and 1 newly synthesized strand. If replication is dispersive, each daughter molecule will have combinations of parental strands and newly synthesized strands. But DNA replication is semi-conservative. Next, we will have to focus on the key enzymes involved in DNA replication one by one. First one is helicase enzyme. Before replication, DNA needs to be unwound. That means both strands of DNA should be separated from one another. So this allows DNA polymerase enzyme to find its substrate and replication can take place. This is done by the enzyme helicase, which breaks the hydrogen bonds and separates the 2 strands of DNA. And next one is Gyrosension enzyme. This enzyme prevents the DNA ahead of the replication fork and removes the tension that is created during unwinding process. And next one is single stranded stabilizing proteins. What these proteins will do is they bind to the single strands thus produced and stabilize them, that means hold them in place. Next one is primase enzyme. This enzyme catalyzes the synthesis of a primer. This is very important in DNA replication. Primer serves as a starting point in DNA replication because the enzyme DNA polymerase, which catalyzes the synthesis of new DNA strand, always requires free 3' hydroxyl group to add new deoxyribonucleotides. So this is provided by the strand RNA strand primer. Primer is a RNA strand synthesized by the enzyme primase. Now DNA polymerase, it catalyzes new DNA strand synthesis. Ligase enzyme joins the DNA strands and fills the gaps. Coming to know the details of DNA polymerase enzyme. This enzyme was discovered by the scientist Arthur Kornberg in 1956, and it directs DNA synthesis in 5' to 3' direction, but it always requires free 3' hydroxyl groups. It adds new nucleotides only to a pre-existing strand. We are now going to talk about 3 types of DNA polymerase enzymes: polymerase 1, 2 and 3. Coming to know their properties. 5' to 3' polymerization activity is very important in the extension of primer. That means adding new nucleotides to the primer. And 5' to 3' exonuclease activity, it removes primer in this 5' to 3' direction. 3' to 5' exonuclease activity provides proofreading function. That means if wrong nucleotides are added to the new strand, they will be detected and removed by this activity of DNA polymerase 1. And next one is DNA polymerase 2 and DNA polymerase 3. Both these enzymes are having 5' to 3' polymerization activity, and 3' to 5' exonuclease activity. 5' to 3' exonuclease activity is absent in these enzymes. It is present only in DNA polymerase 1 enzyme, and this is important in the removal of primer. And DNA polymerase 3 is the main enzyme of DNA replication. Now, let us come to know the steps involved in DNA replication. DNA replication involves 3 steps: initiation, elongation and termination. Coming to know the initiation process. DNA replication always begins at a unique point called origin of replication site, ORIC, and it is a sequence of about 300 base pairs in most of the prokaryotes with highly conserved sequences, and it consists of 3 repeats of 13 base pair sequences and 4 or 5 repeats of 4 base pair sequence sites. They will be recognized by a specific initiation protein complex. First of all, DNA protein binds to this repeat. So this binding facilitates unbinding of DNA, and next DNA protein binds to this repeat, and this facilitates unbinding of DNA because this is helicase enzyme. It breaks the hydrogen bonds and opens the 2 strands so that replication can take place. Next, this is facilitated by DNA protein. Unwinding of DNA strands results in the formation of 2 Y-shaped replication forks in case of bidirectional replication, and these forks move in opposite direction as the replication process proceeds. They move down the DNA, and finally an enzyme primase DNAG catalyzes the synthesis of primer. Primer is a RNA strand. This is the diagrammatic representation showing the formation of initiation complex by DNA and DNA proteins, and this is unwinding of DNA double helix by helicase enzyme. Gyrase enzyme removes the tension stress that is created during unwinding process. Single stranded stabilizing proteins by binding to the single strand stabilize them so that each strand can act as a template for the formation of a new complementary DNA strand. Primase enzyme catalyzes the synthesis of a primer to provide free 3' hydroxyl groups. Next step of DNA replication is elongation. Once the primer has been synthesized by primase enzyme, next DNA polymerase 3 takes the place and adds new deoxyribonucleotides to the free 3' hydroxyl groups of the primer. Here we must know what is meant by leading strand. In case of replication that is taking place on the template strand which is in the 3' to 5' direction, new strand is synthesized continuously in the 5' to 3' direction. So here a single primer is required. In case of replication that is taking place on the template strand which is in the 5' to 3' direction, replication of the second strand is discontinuous. As it is discontinuous, it is called lagging strand. So multiple primers are here required to start each fragment. These discontinuous fragments that are made are called as Okazaki fragments in the name of the discoverer Rizzi Okazaki. So later these short fragments are joined together by the enzyme DNA ligase. That means one strand is synthesized continuously, new strand is synthesized continuously in the same direction of the replication fork movement, that is called leading strand. Whereas the second strand called as lagging strand is synthesized discontinuously in the form of short fragments called Okazaki fragments in the opposite direction of the replication fork movement. By looking at this diagram, you will clearly see how replication is taking place on both the strands simultaneously. One strand is called leading strand because it is synthesized continuously. The second strand is called lagging strand because it is synthesized discontinuously. It is in the opposite direction of the replication fork moment. So multiple primers are synthesized at specific sites because of the non-availability of free 3' hydroxyl groups at the direction of the replication fork moment, and replication is taking place by the enzyme DNA polymerase 3. Next, coming to know the removal of primer by the enzyme DNA polymerase 1 using its 5' to 3' exonuclease activity. After synthesizing the primer by primase enzyme, DNA polymerase 3 adds nucleotides until it reaches the previously Okazaki fragment. There it stops and dissociates. Then this place will be taken by the enzyme DNA polymerase 1. DNA polymerase 1 using its 5' to 3' exonuclease activity removes the primer. Here we get a question, why primer is required for DNA synthesis and later it is removed? Because primer provides free 3' hydroxyl group without which DNA polymerase enzyme cannot initiate new DNA synthesis. But being a RNA strand, it should not be there in the DNA molecule. So it is removed by the DNA polymerase 1 with its 5' to 3' exonuclease activity, and the gaps are sealed by gaps are actually replaced by new DNA strand by the polymerization activity of this enzyme, and then their gaps are sealed by the enzyme DNA ligase. And meanwhile, proofreading is done by 3' to 5' exonuclease activity of this enzyme DNA polymerase 1. So this slide is showing the mode of action of DNA polymerase 1, how it is removing primer RNA nucleotides, and the gaps are sealed by the enzyme DNA ligase. So DNA ligase now seals the gaps by the formation of a phosphodiester bond to form a continuous strand. DNA ligase enzyme ligates the 5' phosphate and 3' hydroxyl groups. This is the end of DNA replication. The replication forks will have to terminate and stop DNA synthesis at a specific point. So this is done at a particular sequence called termination sequence sites. This is just at the opposite side of the origin of replication, and this termination sequence will be recognized by a protein called TUS protein, terminus utilization substance protein. TUS and TER act as a barrier that prevents leading strand and pauses leading strand until lagging strand arrives from opposite direction. So this is done when helicase touches the TUS protein. So this induces termination of DNA. Now the DNA polymerase 3 enzyme synthesizes complementary strands on both sides of the replication fork. This is end of DNA replication. So termination is taking place at TUS and TER sequence. This is the scheme of DNA replication. So it is showing the moment of replication fork and how primers are synthesized and how single stranded stabilizing proteins stabilize the strands. Gyrase enzyme removes topological tension. This is because this is a specific example of the enzyme topoisomerase, and helicase enzyme unwinds the DNA by breaking the hydrogen bonds, and one strand is synthesized continuously here it is requiring a single primer, and another strand is synthesized discontinuously. This is called lagging strand. Here you can see multiple primers are synthesized, and then they will be sealed by the enzyme DNA ligase. Actually, synthesis of lagging strand is a bit later than that of leading strand. So, to get coordination between these two strands, the lagging strand will be pulled out of between the replication fork and DNA polymerase 3 enzyme. Then coming to know what is the result of DNA replication. Replication results in the formation of two identical DNA molecule. Each molecule will have one parental strand and one newly synthesized strand. So, this is actually proving that DNA replication is taking place by semi-conservative mode. So, this is all about DNA replication.
Hello dear students, let me first introduce myself. I am Praveena, working as lecturer in microbiology in Government College for Women, Guntur. Today we are going to learn two important immune disorders, hypersensitivity and autoimmunity. So let us first start with hypersensitivity. So what is hypersensitivity? We know that our immune system is protective in nature by encountering with infectious microorganisms, toxic chemicals and many foreign antigens, and provides immunity to us. But sometimes this immune response it occurs at higher incidence, leading to harmful effects in the host. So we can define hypersensitivity as an exaggerated immune response that results in tissue damage in a sensitized individual on second or subsequent contact with the specific antigen. So in this definition, two terms are important, that is sensitization of the host, and the second one is second or subsequent exposure will result in the hypersensitivity. So what is this in normally this is also called as allergy, and the antigen is termed as allergen. So the first contact with the antigen sensitizes the immune system, and which is called as sensitizing or priming dose, and the second or subsequent contact, subsequent means here whenever he is exposed to the same antigen, it manifests the hypersensitivity reactions. That's why the second dose is called as shocking dose. And basing on the time that is required for appearance of the symptoms and to take the hypersensitivity reactions, these are divided into two types: immediate hypersensitivity and delayed hypersensitivity. So it's a broad classification, and later Peter Gell and Robert Coombs in 1963 they proposed 4 types of classification of hypersensitivity reactions, and this one is accepted universally because it correlates both the process of hypersensitivity reactions and the appearance of the symptoms. So these 4 types of hypersensitivity reactions are type 1 hypersensitivity, which is also called as IgE mediated hypersensitivity, type 2 hypersensitivity are also called as antibody mediated cytotoxic hypersensitivity, and type 3 hypersensitivity known as immune complex mediated hypersensitivity. And the fourth one is cell mediated delayed hypersensitivity. So among the 4 types, the 3 types, that is type 1, 2 and 3, they are mediated through the production of antibodies. Hence it is antibody mediated immune response, and these 3 are immediate hypersensitivity reactions. And the last one is type 4 is mediated through T cells. That's why it is a cell mediated immune response, and it is called as delayed because it will take time from few hours to few days for the appearance of the symptoms and reaction. So first let us see type 1, that is IgE mediated hypersensitivity. So this is induced by those antigen which has the capacity for cross linking of the surface antigen, which surface antibodies, IgE antibodies, which are bound to mast cells and basophils. So on first exposure to the allergen, the B lymphocytes, they are sensitized and they are differentiated into plasma cells and memory cells. The plasma cells in turn produce immunoglobulin E, that is IgE antibodies. On second exposure, the cross bridging of the surface bound IgE antibodies occurs by the antigen, and this leads to the activation and degranulation of the mast cells and basophils, releasing various types of vasoactive mediators. And these mediators, they triggers a series of reactions like smooth muscle contraction, vasodilation, increased vascular permeability and mucus secretion. The type 1 hypersensitive reactions can be once again divided into 2 types. They are anaphylaxis and atopy. Anaphylaxis is a generalized hypersensitivity, whereas atopy is localized hypersensitivity reaction. So first, anaphylaxis, which is an acute, that means it occurs severely and fatal, which can lead to death, and it is systemic, that means it can the symptoms appear all over the body because many organs are affected by this reaction. And the allergens that cause anaphylaxis include antibiotics like penicillins, antitoxins like diphtheria antitoxin, tetanus antitoxin and insect venom. So on first contact, as we already studied that with the first contact, the allergen causes the sensitization and differentiation of B lymphocytes, and the B lymphocytes produce IgE antibodies, which are specifically known as reagin antibodies. And these antibodies binds to the Fc receptors of the mast cells or basophils and sensitizes these cells. So on second or subsequent exposure, the allergen it causes the cross bridging of the adjacent IgE on sensitized cells, and this leads to activation and degranulation of the mast cells and basophils, releasing various types of chemicals like histamine, serotonin, heparin, leukotrienes, platelet activating factor and eosinophil chemotactic factor A and prostaglandins. So here you can see the process of type 1 hypersensitivity, where the antigens are when exposed first time, on first exposure the antigen cause the differentiation of B cells into plasma cells and memory cells, and the plasma cells, they produce specific IgE antibodies, and these IgE antibodies they are present on the surface of the B lymphocytes. And on the second exposure, whenever the allergen enter the immune system, the allergen cause the cross linking of the IgE antibodies, leading to degranulation of the mast cells, releasing all the chemicals from the mast cells, which cause the specific symptoms of the anaphylaxis. So in anaphylaxis, the reactions include smooth muscle contraction, vasodilation, that is dilation of the blood vessels, and increased vascular permeability. So because of all these reactions, it will lead to respiratory impairment, because the smooth muscle contraction, the muscle contraction of the bronchioles, and BP is reduced due to the vasodilation, and rapid loss of fluids enter into the tissue spaces, and the individual dies within minutes due to asphyxiation, that is reduction of the oxygen and reduction of blood pressure, and finally due to circulatory shock. That's why anaphylaxis is a fatal type 1 hypersensitivity reaction. And the second type is atopy. It is a chronic and recurrent and non-fatal. Typically it is localized to a particular organ, and the symptoms mainly includes depending upon the route of entry of the allergen. So the allergens includes inhalants such as pollen grains, fungal spores, animal dander, and house dust mites, and ingestants like eggs, milk, fruits, and fish. And the symptoms I have already told that they depends upon the route of entry of the allergen. So the most common examples for atopic reactions include hay fever, bronchial asthma, allergic rhinitis, and food allergies. So here hay fever, it involves the upper respiratory tract, which is characterized by congested nasal passage, coughing, sneezing, and fever. And the second one is bronchial asthma, which is the atopic reaction of lower respiratory tract, and the symptoms mainly includes filling of the alveoli with fluids, and construction the bronchial walls of bronchioles, they will be narrowed, due to which he feels very difficulty in breathing, and the characteristic symptom of bronchial asthma is wheezing sound during the exhalation. In Telugu we will call it as pilli kootalu. And the third one, allergic rhinitis, so which is very common nowadays in majority of population because of the increased pollution. And here this allergic rhinitis, the symptoms include coughing, sneezing, and congested nasal passage, and sometimes allergic sinusitis, and this occurs mainly when the person is exposed to all the inhalants, allergic inhalants, are cool climate and strong smells like that. And food allergies, this food allergies mainly, these are the atopic hypersensitivity reactions that occur due to the ingestion of allergic foods like fruits, eggs, milk, like that. When the person become allergic to these food items, he will show symptoms from simple hives. Here you can see the red spots, hives, to urticaria and sometimes severe eczema. So here we will stop for a few seconds, and next we will proceed with the type 2 hypersensitivity reactions. Okay, now we will continue with the next hypersensitivity reaction, that is type 2 hypersensitivity reaction, which is also called as antibody dependent cytotoxic or cytolytic hypersensitivity reaction, because it will lead to the destruction of the cells and tissues. Here the antibodies involved are IgG or IgM, and these are directed against antigens that are present on the cells or tissue surfaces. So these antibodies, when they combine with the cell surface antigen, they activate the complement and bind to the effector cells through Fc regions and stimulate the complement pathway. Complement pathway causing the destruction of the cells. So, examples for this type 2 hypersensitivity reactions are erythroblastosis fetalis and ABO blood transfusion. Erythroblastosis fetalis, it is a anemic disease of newborn which causes hemolytic anemia. So, this occurs when a when Rh negative mother carries Rh positive fetus in her womb. So, here because of the mismatching of the Rh antigens of the mother and fetus, during the pregnancy the fetal blood will come into the maternal circulation, and the Rh antigens are exposed to mother blood circulation. As a result, the immune system of mother produce anti-Rh antibodies. So, here in the first delivery, the sensitization of the mother occurs against the Rh negative antigens, and it doesn't cause any harm to the first baby. But later on the second delivery or on subsequent deliveries where the mother carries the Rh positive fetus or baby, during the pregnancy, as already the anti-Rh antibodies are there in maternal circulation, they will attack the Rh antigens of the fetus, leading to hemolytic anemia and results in miscarriages, that is abortions. And the ABO blood transfusion is another example for type 2 hypersensitivity, where the mismatching of blood groups occurs between the donor and recipients, where the red blood cells will be destructed and it becomes fatal to the individual. Next, the type 3 hypersensitivity reaction, which is also called as immune complex mediated hypersensitivity. Here immune complex means the antigen and antibody complex, and generally these antigen antibody complexes, they are removed very effectively by the monocytes of the reticuloendothelial system. But sometimes these immune complexes, they are not effectively removed and they will deposit on the basement membranes of many organs, leading to type 3 hypersensitivity reactions. So, because of the deposition of these immune complexes on the epithelial cell basement membranes, they will activate the complement, especially the C3A and C50, which are also called as anaphylotoxins, and activates and cause the degranulation of the mast cells, basophils and platelets, releasing histamines and other chemical mediators. So, this triggers a variety of inflammatory responses like increased vascular permeability, release of oxygen containing free radicals and proteolytic enzymes from cytoplasmic granules, which cause the damage to the blood vessels, and the platelet aggregates in the aggregate forms on the vessel walls, and chemotactic factors from various complement components attracts the effector cells, neutrophils, and all these are activated and triggers a variety of inflammatory reactions. So, here we can see in the picture the immune complexes, they are deposited on the epithelial basement membranes of the blood vessels, and they attract the platelets, becomes aggravated, forming the microthrombi formation, and they cause the complement activation. As a result, so many mediators are produced like chemotactic factors, which cause the flowing of polymorphonuclear leukocytes, that is neutrophils, and all these cells towards the towards the site where the immune complexes are deposited, and cause the vasculitis, that is the inflammation of blood vessels. So, the type 3 hypersensitivity reaction is two types. One is Arthur's reaction and the second one is serum sickness. Arthur's reaction, it is actually an experimental reaction, and it was done by Nicholas Maris Arthurs in 1903, and it is a localized hypersensitivity reaction, and it occurs when large amounts of horse serum is introduced into the rabbits repeatedly. The rabbits develops this type 3 hypersensitivity reaction, which is characterized by necrotic vasculitis and edema. And the second one is serum sickness, which is very acute and systemic, means it involves so many organs of the body, host body. So here the antigens like penicillin or heterologous serums like diphtheria antitoxins or tetanus antitoxins like foreign materials, when they are introduced into the host, the antigens combines with the antibody, forming systemic tissue deposits, and the reaction occurs between 5 days to 3 weeks, and the reaction is characterized by fever, dermatitis, lymphadenopathy, arthritis, that is inflammation of the joints, glomerulonephritis, that is inflammation of the walls of kidneys, nephrotic system, and endocarditis, where the endocardium becomes inflamed in heart. So because here many vital organs are involved in this reaction, it is sometimes very fatal reaction. Okay. And the specificity of this serum sickness is a single dose of antigen can act as both sensitive as well as shocking dose. So generally what happens, first the host has to be exposed for the first time, which we called as sensitizing dose, and second time the reaction will appear, which we call it as a shocking dose. But in serum sickness, the single dose acts as both sensitive as well as shocking dose. Now let us see the type 4 hypersensitivity reaction, which is called as cell mediated delayed hypersensitivity. So the name itself indicates it is mediated through T cells, and here the T cells, the subset of T cells which are called as TDTH cells, which stands for delayed type of hypersensitivity T cells, are involved, and the symptoms takes very long time to appear. That's why it is called as delayed hypersensitivity. So on contact with the specific antigen, it causes release of lymphocytes, macrophages and basophils to the affected tissue and result in extensive tissue damage. And this type 4 hypersensitivity is two types. One is infection type and the second one is allergic contact dermatitis. So here we can see the antigens, they stimulate the lymphocytes producing T cells, that is T lymphocytes, and they in turn produce so many cytokines which activates the macrophages and mast cells, all these neutrophils, all these, and leading to cellular infiltrate. So the first type of type 4 hypersensitivity reaction is infection type, where and actually it was first time it was observed by Robert Koch. So when he injected the tuberculin protein isolated from Mycobacterium tuberculosis, so he took the tuberculin protein, which is a partially purified protein, and injected into intradermally on the forearm of the individual, and the reaction begins in 8 hours and results in reddened area at the site of injection, and the induration reaches in its peaks within 12 to 24 hours, within 12 to 24 hours, and it reaches the peak in 48 hours and then subsides in a tuberculin positive individual. That means here the test is mainly done to know whether the person is previously exposed to Mycobacterium tuberculosis. So if the person is exposed to Mycobacterium tuberculosis, he will sensitized by the tuberculin protein, and when we inject the tuberculin protein externally, already because he is sensitized, he will show the type 4 hypersensitivity reaction. So this, and the tuberculin test will be negative in tuberculin negative individuals, which shows that he is not previously exposed to Mycobacterium tuberculosis. So type 4 hypersensitivity reaction develops in many infections with bacteria and viruses, and even also the parasites, especially when these parasites are intracellular, like that. And this infection type or the tuberculin type test is also done to know whether the person is previously infected with diseases such as leprosy, brucellosis, candidiosis and herpes simplex virus. And the second type of type 4 hypersensitivity is contact dermatitis, and this can be observed in a large population when the skin is exposed to certain antigens. So this is actually an eczematous skin disease. So here you can see there are the rash which appears like boils filled with fluid. So the characteristic feature of this eczematous disease is rash and the weeping boils. So on contact with the allergen first it combines with the proteins of the skin which are called as haptens, and they activates the T cells, and mainly the allergens includes proteins, chemicals like dinitrobenzene, picryl chloride, certain dyes, and sometimes penicillin, drugs like penicillins, and antibiotic topical ointments, and nickel coated jewelry, cosmetics and plant materials. So this is about hypersensitivity, the various types of hypersensitivity, all this. And now let us see what is the treatment for these hypersensitivity reactions. So the treatment generally includes, the first and foremost is we have to avoid the contact with the allergen, and second thing is the medicinal practitioners will do the immunotherapy, where they will inject increased doses of allergens so that the individual immune system will shift from production of IgE to IgG. And the second one is by administering anti-IgE antibodies, so that these antibodies will block the IgE immunoglobulins. And like drugs like antihistamines and epinephrines are also used in treatment. So this is about hypersensitivity reactions. Okay students, now we will see the second type of very important immune disorder, that is autoimmunity. The name autoimmunity contains the prefix auto, means self. So here the immune system responds to self antigens instead of non-self or foreign antigens, and this occurs due to the lack of discrimination between self and non-self antigens by the immune system. So this causes a serious damage to the cells or organs, and sometimes lead to fatal consequences, which we call them as autoimmune diseases. And these autoimmune diseases, they are mediated through either antibodies or sensitized T cells. So the autoimmunity can be described using this comic picture, where this is the antigen and this is the self antigen, which is saying that I am yours, that is I am part of your body, I am not a foreign thing for you. But the cells of the immune system, they are mistaken it as a foreign antigen or they are non-self antigen, and they will react to the self antigen. So due to this loss of discrimination by the immune system between self and non-self antigens, the autoimmune disorders are also called as autoimmune diseases occur. So what are the causes of autoimmunity? So the first cause is the sequestrated antigens. So sequestrated antigens means these are the antigens which do not come under blood circulation during the development of immune system. So later, whenever these antigens come in blood circulation in the individual life, the immune system will consider them as foreign and they will attack the antigens. So and cause the disease, autoimmune disease. And the examples for the sequestrated antigens includes lens proteins, spermatozoa, all these. And sometimes even the fetus for a pregnant lady is also considered as a non-self thing that will result in that very medical care, that needs the medical care. And the second thing is molecular mimicry. Molecular mimicry means here the foreign antigens like certain bacteria or viruses, they share same structural similarity with the individual's self components. So because of this structural similarity between the foreign antigens and the individual self components, it will leads to autoimmunity. For example, the M protein of Streptococci pyogenes, they share same structure with that of glycoproteins of heart and joints, and this leads to the heart failure during rheumatic fevers. And certain bacterial and viral infections are also responsible for autoimmune diseases. So what are the factors that influence the development of autoimmunity? The first important thing is genetic factors, because the autoimmune diseases, they run in families due to the major histocompatibility complex, which is the major protein in immune response. It is coded by the genes. So genetic factors, they play a very important role in autoimmune diseases. And viral infections, they sometimes cause the autoimmune diseases. And hormones, especially the sex hormones, it is found that the female sex hormones like estrogen, they enhance or they activate the autoimmune diseases, whereas the male hormones, androgens, they suppress the autoimmune diseases. And this is the reason which can explain why women develop more autoimmune diseases when compared with the male. And the last thing is the psychoneuroimmunological factors, which includes the stress and neurochemicals, the release of various neurochemicals like serotonin, acetylcholine, all these, they also sometimes leads to autoimmune diseases. So the autoimmune diseases can be divided into two types. One is organ specific autoimmune diseases, and the second one is systemic autoimmune diseases. So what are organ specific autoimmune diseases? Here the immune response is directed to specific antigens that are present on the tissues or organs, and through an antibody mediated immunity or cell mediated immunity. And due to this, the function of that organ is blocked because of the autoantibodies, and the symptoms are also limited to that particular organ. So because the antigen involved are related with only a single organ, and the symptoms are also restricted to that single organ, these disorders are called as autoimmune disorders or autoimmune diseases. So let's have a glance on what are these autoimmune disorders or diseases. So the first, some of the examples we have, the first one is Addison's disease, where the autoantibodies are produced against the adrenal cells, and the symptoms includes hypocorticolism, that is the reduced production of adrenal hormones. And the second one is Gould-Pasteur syndrome, where the autoantibodies are produced against collagen of the basement membranes of kidneys and lungs, and as a result they will show very fatal symptoms like kidney failure, pulmonary hemorrhage, that is bleeding from the lungs, and often it leads to death. And multiple sclerosis, where the autoantibodies are produced against brain or white matter, and the symptoms includes double vision, blindness in one eye, muscle weakness, and also trouble in sensation. And rheumatoid arthritis, where the Fc portion of the antigen is the autoantibodies are called as RA factor is produced against the Fc portion of the antigen, that is immunoglobulin G, in connective tissue, which leads to the inflammation of joints and pains. Actually, these are the systemic autoimmune disorders where the entire body will be affected. And the next one is systemic lupus erythematosus, where the autoantibodies are produced against double-stranded DNA and the nuclear proteins, which leads to fever, weakness in arthritis, and skin rash. And the fourth one is scleroderma, where the autoantibodies are produced against nuclei and lungs, which leads to swelling in limbs, sclerodactyly, that is because of the more amount of production of the collagen, the skin and the connective tissue, they become very hard and thickened. Due to this, they are unable to move their fingers and toes. And Raynaud's disease, where the blood vessels in the hands and feet, they becomes very narrow, and they feel very discomfort in blood. Due to the discomfort in blood circulation, they feel numbness and pain in hands and feet. So these three are systemic autoimmune disorders, that is multiple sclerosis, rheumatoid arthritis, and systemic lupus erythematosus, scleroderma. These are the systemic autoimmune disorders which affects many parts of the body, and sometimes the people, once they are encountered with the autoimmune diseases, they have to face the symptoms for lifetime. So here you can see the pictures of two important systemic autoimmune disorders, that is systemic lupus erythematosus and scleroderma. So here various organs like lungs, heart are affected, and the inflammation of lungs occurs, heart occurs, and the characteristic features of this systemic lupus erythematosus is butterfly rash, that where the rash, you can see the rash, it starts from the nose and it spreads through the cheeks in the form of, in shape of butterfly. That's why it is called as butterfly rash. And the second one is scleroderma. So here you can see the sclerodactyly, where the fingers, they become very stiffened due to the increased production of collagen, and here the rash all over the skin. So these actually these are the very serious symptoms that appears in these two systemic autoimmune diseases. Then what is the treatment for these autoimmune diseases? The first thing is to administer non-steroid anti-inflammatory drugs, and second one is taking supplements like thyroxine or vitamin B12. So if the person is suffering from hypothyroidism, he will be given thyroxine supplements, and if the person is suffering from pernicious anemia, he will be given vitamin B12 supplements like that. And the third most is by giving immunosuppressant drugs. So these drugs, they suppress the immune response so that they will feel some relief from the symptoms, which includes peptide drugs, which blocks the MHC molecules. As we have already learned, MHC molecule plays a very vital role in autoimmune diseases, and giving monoclonal antibodies against certain immune cells that are involved in immune response, like CD4 cells, interleukin 2 receptors and T cell receptors. So with this I am concluding here, and thank you students for your patient listening.
Students, we are going to take up a very important topic related to the 6th semester chemistry elective paper. As you all know, under 6th semester, under CBCS system, you will be having one elective paper you have to choose from each subject. The topic, the 7th A and 7th B, there are two papers that are included under APSHA syllabus. So the first paper is the 7th A paper, which we have discussed earlier. That is nothing but the separation methods and the instrumentation techniques. And the 7th B paper is the environmental chemistry, 7th B. So for the benefit of the students who choose this environmental chemistry paper, we have taken up this particular module. Under the first module, I am going to give a brief introduction related to the environmental chemistry. What is environment and what is environmental chemistry, and what are the different type of topics you are going to take up under this unit. As you can see in the slide, the first unit of your syllabus is, as you can see, concept of environmental chemistry, scope and importance of environment in nowadays. And the second one is the nomenclature of environmental chemistry, segments of environment and natural resources, renewable resources, solar and biomass energy and non-renewable resources, thermal power and atomic energy. The last, reactions of atomic oxygen and hydrological cycle. And of course, you will be having all these topics under first unit. I am going to take up as much as possible in this from this unit. Let us take up the environmental chemistry, but before actually going into the environmental chemistry, let us take up the definition of environment. What is environment? Environment, as you all know, it is nothing but